Okay, hello people. It's a great pleasure to introduce Dr. Ammar Al-Chalabi, MB, CHB, and PhD. Dr. Al-Chalabi, thank you for having accepted our invitation and be here today participating in the 6th ALS Symposium of the Pro-Cura da Ela. Last year, unfortunately, we couldn't have your presence here due to the pandemic. But today, we are very happy to be with you in our online symposium, okay? Thank you for taking your time to be with us here. And now I will read your biography a little bit. Okay, Dr. Ammar Al-Chalabi is a neurologist and scientist, professor of neurology and complex disease genetics, director of King's Motor Neuron Disease Care and Research Center in London. His research focuses on amyotrophic lateral sclerosis. As professor of neurology and complex disease genetics, Dr. Ammar Al-Chalabi has been at the forefront of MND research for over 25 years. One of the most recognizable and charismatic researchers in MND, he has built relationships globally throughout his career that have vastly increased the scope of what MND research can achieve. His research team works on finding the causes of ALS and what might influence the way the disease manifests and progresses, particularly where this involves genes, environmental exposures, or changes in thinking and coping. Dr. Al-Chalabi will talk now about the epidemiology of ALS, the relationship between genetic and environmental risk factors. Now, I give you the floor, Dr. Chalabi.
Thank you very much for that really lovely introduction. I will share my screen so that you can see it. So I hope you can now see my screen.
Yes.
Okay, so I'm going to be talking, as you said, about the epidemiology of ALS, genetic and environmental risk factors. Now, there are two obvious causes of ALS. The first is family history, which is present in about 5 to 10% of people, and tells us that there must be a genetic basis for ALS. We also know that the risk increases as people get older, which tells us something about the mechanism must be related to aging. Here you can see a graph of the risk of ALS in a lifetime as somebody ages. And you can see that the risk keeps rising as people age, but also that males always have a stronger risk than females. That explains why ALS occurs more frequently in men than in women. A very striking feature is that the lifetime risk of ALS is about 1 in 300. So it's quite common in terms of a risk over a lifetime, but that's usually hidden because we only talk about incidence or prevalence. The prevalence is very low because the lifetime of a person is shortened if they have ALS significantly, which means there aren't many people with ALS at any one time. Now we know that, as I said, about 10% of people have a family history of ALS. And in those people, we can often find genes that have a mutation in them, a variation, which increases the risk of ALS. Here you can see a graph showing the discovery of genes with time on the x-axis and the number of genes found on the y-axis. The size of each gene, the size of each circle tells you how important that gene is for ALS based on publication. So the very big circle in the middle is from a gene called C9orf72, and mutation in that gene affects about 10% of people in some parts of the world. You can also see some of the spheres are red, some are blue, and some are both. The red indicates that that variant is only found in people without a family history, and the blue is for people found with a family history. So you can see most gene variations are found even in people without a family history or with a family history. The labels for the genes are grouped into 3. The green labels indicate genes that are involved in protein homeostasis. The purple labels are for those genes involved in altered RNA binding, so something to do with RNA processing. And the orange are those involved in the cytoskeleton, in maintaining the shape of a neuron. But although those genes have a large effect, most genes only have a small effect. So this is what's called a genome-wide association study. Every single dot here represents a test at a particular gene variation across the genome. And in this graph, there are something like 7 million dots. You can see the red spike in the middle corresponds to the C9orf72 gene that I just mentioned. Everything in red is a gene where variations slightly increases your risk of ALS, or in the case of C9, actually greatly increases your risk. Now, this study was done with about 12,000 samples from people with ALS, but more recently, we've done a study with nearly 30,000 people with ALS, and you can see we discover many more genes, and some of the gene variations we found before become much more prominent. But this tells us that lots of gene variations might slightly increase your risk of ALS. So probably if you have 5 or 6 of these gene variations, each one only increases your risk by 5%, 10%. If you have enough of them, it will nudge you very strongly towards developing ALS. These particular 3 genes are found in people with a family history of ALS, SOD1, C9orf72, and NEC1. But they also come up on this test in people without a family history. And these 3 genes have variation that is also found causing other diseases than ALS. C9orf72 is found in a condition called frontotemporal dementia. The same with UNC13A on the right. And on the left, the gene MOBP, MOBP, variation in that gene is found in a variation of Parkinson's disease called progressive supranuclear palsy. So that tells us that having a disease gene for ALS doesn't necessarily mean that you will develop ALS. You may develop a different condition, or you may not develop ALS at all, but it increases your risk. We can use these genetic studies to also examine the possibility of environmental risks. So we've used some techniques. One of them is called Mendelian randomization. In this method, we can find things that cause changes that cause a disease. So here we've tested whether cholesterol change can cause ALS, and we showed that a high cholesterol is implicated in ALS. That's high total cholesterol, seems to increase the risk of ALS. We can also show from another related test called epigenetics testing that actually it's the HDL component of cholesterol, the high HDL, which is what increases the risk. Similarly, having low triglycerides and being slim also increase the risk. So this is a little unusual. It tells us that if you are protected from a risk of heart disease, you seem to have an increased risk of ALS. Interestingly, glucose intolerance, which does increase your risk of heart disease, it's a form of diabetes, is protective in this study. Now, all of this work needs to be confirmed in other studies, but it's a demonstration that we can use genetics to try and identify environmental risk factors. We can also just ask people, what have you done in your life, and give them a questionnaire to complete and see if that works. The problem with that kind of technique is that people may not remember very easily what they have done many years ago. And some of the questions we might need to ask might relate to childhood, when the person will have no recall effectively of what they were exposed to. Despite that, people have tried, but there are no strong reproducible associations with any job, place, lifestyle, or life events. But there does, in some studies, seem to be an increased risk from military service, from smoking, although more recent studies cast doubt on that, from exercise, but again, that doesn't mean that exercise causes ALS. It may just be that if you are built for exercise, if you have a very graceful motor system, that maybe that increases your risk. It might be the way you're built. And then life events such as infection, electric shock, and physical trauma have all come up in some studies, but not in others. So the environmental risk factors are very difficult to definitely identify. So smoking, about 40 studies have been performed. About half of those show a strong link, and about half of them show no link. The most recent study, which uses the technique Mendelian randomization and studied about 30,000 people, using genetic techniques found no link between smoking and ALS. That doesn't mean there isn't a link, but so far there's no strong evidence of a link, and that's the largest study done so far. Heavy metals do seem to increase your risk. So cadmium and lead exposure do seem to increase the risk of ALS, while zinc exposure seems to decrease the risk. We can also use other techniques to try and find what environmental factors might cause ALS. In this study, we've plotted the location of every single person with ALS over a 10-year period in the southeast of the UK. What you can see is some clusters. I've highlighted the ones I see in red. So you can see there's quite a lot of clusters around the coast, and there's a very big cluster in the centre in London. But of course, in London, there are more people, and therefore you will have more cases. And in the UK, people, when they retire, tend to move to the coast because it's a nicer place to live and it's warmer. And remember that ALS is a disease where the risk increases as you age. And so if you have an older population, you're more likely to have ALS at the coast. So these kinds of studies are very difficult in terms of revealing what environmental risk factors there might be based on clustering. But we can use them and we can gain some information from them. Now, one very clear question is whether viruses might cause ALS. We know that poliovirus causes a motor neuron disease. And so it's sensible to ask, do other viruses cause ALS, or even perhaps poliovirus? Now, a study we performed back in the 1990s, which was then published in 2000, looked at reverse transcriptase in the blood. Reverse transcriptase is an enzyme released by viruses, some viruses called retroviruses. And we found that nearly 60% of people with ALS had reverse transcriptase detectable in their blood. But when we looked at the general population, only 5% of the general population had the same signal. So that might suggest that a virus, a retrovirus, causes ALS. Now, we know that ALS doesn't appear to be infective. So that's a slight difficulty in understanding why we would see this signal. We also showed that relatives of somebody with ALS, also about 60% of those had this signal, whereas the wives and husbands of people with ALS, the spouses, had the 5% level. They had the same as the general population. So that tells us that this isn't an infective virus. And in fact, there is a group of viruses called endogenous retroviruses, which are genetic sequences that are part of the human DNA. And those gene sequences can be activated in some conditions. So we think that probably in ALS, this gene sequence is activated. Whether that's a result of the disease or a cause of the disease is still under investigation. The important thing is that this is not an infective cause of ALS. It's an inherited cause. And that it's very common. Everybody carries these retroviruses. There's nothing special about the person with ALS in terms of these sequences. But for some reason, they seem to be activated. So these viruses are embedded in human DNA through evolution. They're in the same group as HIV, and I stress again, it is not infective. Everybody carries these gene sequences. They account for about 8% of our genetics. Now you have to compare that with how much of the human genome codes for protein, codes for the proteins that make you or me, and that's about 1%. So much more of our DNA codes for these viruses than codes for proteins that make us. And these viruses have actually become important for things like placenta formation. They're an essential part of the way humans are built. A more recent study has suggested that one of these sequences called HERV-K is possibly the culprit, is possibly the cause of ALS. And we now have a phase 3 clinical trial looking at a drug that might be a treatment for this to see whether that would work. Inflammation has also been implicated in ALS and actually in other neurodegenerative diseases. If we look post-mortem, there is evidence of inflammation with infiltration of macrophages and mast cells into the cells, into the nervous system, and also immunoglobulin and complement, which are two components of the immune system, are deposited in the nervous system. And there are some genes, like there's one gene called TLR4, which signals the immune system to activate. That seems to be switched on in people with ALS. And also other immune cells like microglia, which are cells that live in the nervous system and defend it. Those seem to be activated. And also another type of cell called the T regulatory lymphocyte, or a TREG, is also decreased in ALS. And that cell is important in regulating the immune system. So all of this evidence suggests that a problem with the immune system may underlie ALS. Again, it doesn't mean it causes ALS. It may be that when you develop ALS, the immune system changes, but it may be that when the immune system changes, that causes ALS. So we still have to disentangle which direction happens, but we've seen this signal that the immune system is somehow important. There are a number of mysteries about ALS that remain difficult to explain. We know that if you have an ALS gene, as I showed you earlier, some of those genes don't cause ALS, they cause other diseases. And also, you can carry an ALS gene and never develop ALS. Why is that? Also, you've seen it's difficult to find environmental causes of ALS. Why? It's not difficult to find environmental causes of cancer, for example. There's also the problem of time. People with a gene cause for their ALS are born with that genetic change, but they don't develop ALS until they're 50, 60, 70 or older. Why? How can you carry this change all your life and not develop the disease? And why isn't there a single cause of ALS? And why, again, do these genes switch on as you get older rather than younger? So we can look to cancer because all these problems are similar. It's the same kind of questions you could ask about cancer. Now, cancer, we know, is a multi-step process. For example, you may be born with some genetic causes. If you smoke, that might damage your DNA. You might then be infected by a virus, have some kind of inflammation. You do something else like drinking coffee, which activates your cells. But unless you have all of these steps, you won't develop cancer. Somebody who is exposed to all of these steps will develop cancer. So we asked, is that the same in ALS? Now, you can detect multi-step diseases like this using some mathematics. A disease that that has a multi-step process underlying it obeys particular mathematical rules that show a power relationship between the incidence of the disease and the age of onset of the disease. So that as you age, your risk of the disease, the rate of the disease, increases to a power. So it may be squared or cubed, but you can work out the mathematical relationship between the two. That means if we plot the log of the incidence of ALS against the log of the age and see a straight line, then ALS is also a multi-step process. If we don't see a straight line, then ALS isn't a multi-step process. If we do see a straight line, then the slope of the line tells us about the number of steps because of the way the mathematics works. So we asked this question in our register of the Southeast of England. So the Southeast England ALS register, the SEALS register, this is a plot of the log of incidence against the log of age for our patients, and you can see it obeys a straight line. So the closer it is to a straight line, the higher the R squared value is, the closer it is to 1. And a value of 0.95 is suggesting a very good relationship to a straight line. We then went to our colleagues in the Netherlands, and their results also show a straight line. So that again suggests that ALS looks like a multi-step process. So it's not something you just get or you don't get. There are a number of steps you have to go through before the disease starts. To confirm this, we went to colleagues in Italy, in Ireland, and in Scotland, and we showed the same thing in every single register. When we combined them, the straight line effect is even stronger, which tells you they all have the same slope. In other words, they're all telling you the same number of steps is required for ALS. And when you do the maths, it's actually, the slope is 5, which means that ALS is a 6-step process. It's always one more than the slope. So there are 6 steps required to develop ALS. Since we did that study, it's been replicated multiple times across the world, in every single time showing a 6-step process, except for one study that suggests a 5-step process. Now you can take that further and ask, what are these steps? One way you can do that is to take out people who just have a genetic cause, for example, and see what does that do to the number of steps required. And when we do that, we can show that some of the genes account for more than one step. Particularly, the SOD1 gene accounts for 4 of the 6 steps. So if you have mutation in SOD1, you've already got 4 of the steps accounted for. You only have to be exposed to things that cause the last 2 steps to develop ALS. So that explains also why some genes have a large effect. if they use up many of those steps, it's easier to develop ALS for that person than if you don't have those steps used up. You can also ask the question of whether lifestyle changes the slope. This is a study from Naomi Ray in Australia that showed that if you have pre-existing heart disease, that seems to increase your risk. That is against what our other study showed about the environment, but that just shows you how difficult it is to disentangle environmental risk factors for ALS. You can use similar studies to show that type 2 diabetes might be protective, and that also fits with our genetic work, but other studies do show that it might increase risk, so that's still under investigation. But the important thing is, these multi-step models do answer all of these ALS mysteries. For example, the fact that ALS genes cause other diseases is because the exact steps might differ at one or more points. So if you go 1, 2, 3, 4, 5, 6, you develop ALS, but if you go through steps 1, 2, 3, 4, 5, 7, a different last step, maybe you develop frontotemporal dementia. Not all gene carriers will develop ALS because unless you have the last steps, you won't develop it. So you could carry the gene, but never be exposed to the things that will push you to develop ALS. Environmental causes are difficult to find because different factors might trigger the same step. In the same way as smoking or exposure to X-rays both cause mutation, it might be that many different things cause some of the steps for ALS. The increasing risk with age is because it takes time to accumulate all the steps, and there isn't a single cause because different factors can trigger the same steps. And the same for the age-dependent gene penetrance. It just takes time to accumulate the steps. So this model explains these mysteries quite well. We can also think of ALS like this, that you're born with a particular genetic predisposition, which may be very low or very high. As you age, your cells are damaged with time, and you're also exposed to various environmental risk factors. But unless you reach a certain level, you won't develop ALS. So genes, time, environment. If you do reach that critical threshold with the burden of exposures you've had, then you'll develop ALS and you have a self-perpetuating process. You could put the multi-steps at the bottom. So those are your 1, 2, 3, 4, 5, 6 steps, and then you develop disease. And you can imagine some people are born with a lot of genetic load, so maybe 3 or more steps are accounted for. Some people might have an inherited single gene that accounts for many steps. But the important thing you can detect from this is if you do have a large genetic cause of your ALS, then it should be easier for us to find the environmental risks in those people because they have fewer steps to go through. So it should be a more targeted finding. The multi-step model happens to occur in nature in a model of ALS. So there's a mouse that develops ALS. That mouse has endogenous retrovirus in the same way as humans have endogenous retroviruses, these gene sequences of viruses. The mice have them as well. In fact, all mammals do. Some mice have many copies and others have only a few. If you have a mouse with many copies, some of those mice also have genetic variations that switch on that retrovirus or switch it off. So you might be a mouse that has many copies of this retrovirus and a genetic variation that switches it on, that activates it. If you're one of those mice and you're infected with another virus called LDV virus, then you will develop ALS. This is a natural model of mouse. So you can see this mouse has a 3-step process. It has a combination of a genetic risk in the endogenous retrovirus, and it has an environmental risk from the LDV virus. And it needs the multiple steps before it develops disease. So you can imagine a similar process in humans. The other thing that the multi-step model tells us is that genes that increase risk should lower your age of onset, because your risk will be higher at any age and the age of onset would therefore be lower. So we asked that question. We looked at how people with familial ALS and compared them with the people with no family history. And we showed that the people with familial ALS have an age of onset about 5 years younger than the people with apparently sporadic ALS. Now, that's not really a fair study because some of the people with sporadic ALS have a genetic cause. I showed you that at the beginning. If you have a genetic cause for your ALS or a non-genetic cause for your ALS and you have no family history, that is a fairer comparison. And if you compare those two groups, you can again show that even in the people with no family history, those with a genetic cause have a younger age of onset. And if you compare the people with no family history but a genetic cause with the people with a family history and a genetic cause, there's really no difference in their age of onset. So that shows you that it's the genes themselves that are lowering the age of onset. So for some reason, if you have a genetic risk, that lowers your age. Well, that's what we would expect from the multi-step model. So again, it's just evidence that this multi-step process is underlying ALS. But it leads us to a question. Will pathway or gene-based treatments be more effective in younger people? In other words, if we have a treatment that's developed because of knowledge of these genetic pathways, maybe it'll be more likely to work in people who are younger because they're the people where these pathways are more affected. Now, we don't know if that's true or not, but it's a question that's important to ask. We can also ask it the other way around and say, if we see a young person, are they more likely to have a genetic cause? And this is their probability. So this graph shows that the younger you are, the more likely you are to have a genetic cause of your ALS. And therefore, it's more important to test younger people, particularly for genetic factors. So we've got all of this genetic and environmental information. Can we use it to work out the mechanism of ALS, the pathway? Well, many mutations lead to a gain of function. And that doesn't really tell us about the pathway because the gene hasn't lost its original function. So for example, if there's a gene involved in like the dismutase gene, for example, superoxide dismutase, if it's involved in changing the oxygen status of a chemical, but it's actually gained a new function, unless we know what that new function is, we don't really benefit from knowing what the gene did. So we have to know what the new function is to really understand the pathway, or find a gene where losing the function is what matters. So the ones where we lose a function are recessive genes, or those involved in the skeleton of the cell, for example. But we can build pathways like this. So in this particular diagram, you can see we've put some genes that are all involved in protein homeostasis. You can see the different parts of the pathway that would be involved. So you can imagine building a model of ALS like this, where we say, well, these proteins are important here, and this is how it all fits together. And then you might be able to make a treatment that targets part of this pathway and repairs it in some way. But another question we can ask is whether we should care what causes ALS. Our patients don't come to us before they have ALS. They come to us once they have ALS. So the risk genes have already done their work. What we need to know is, what is continuing the process? Why do some people live a long time with ALS and some people don't? What is that difference? Are the genes that increase your risk also the ones that shorten your survival? Or are the two things completely separate? Or perhaps it's a mix. For some causes, the two things are connected, and for other causes, they're completely separate. So we've done a genetic study. Again, this is like the ones I showed you before. Every dot is testing a different genetic variation to look to see whether any gene variations affect the survival of somebody with ALS. And we can find some genes that might do. So CAMTA1, for example, binds calcium, and the IDE gene is involved in the insulin pathway. So that's interesting because I showed you the link with type 2 diabetes might be important. But the important thing is these aren't risk genes. So this is an example where the genes that affect the progression of the disease are separate from the genes that are increasing risk. But some genes do increase both. So for example, the C9orf72 gene, if you have a variation in that increases your risk of ALS, it also gives you a worse outcome on average. It makes a more rapid disease. The same for the UNC13A gene. Now, this is a graph called a Cox regression or a Kaplan-Meier graph. So it's a survival graph. On the y-axis is the percentage of people alive at any time point. And on the x-axis is the time. If a population of people dies rapidly, then the curve will be very steep. If they live a longer time, the curve will be flatter and more to the right. And you can see that the people with the green, the light blue color, which are the non-carriers, so they don't carry a genetic cause, have a better survival than the other two lines, which are people carrying these disease genes for their ALS. This study was done in, this was a genetic study combining different clinical trials where we also had genetic data. Now, in those clinical trials, they were studying whether lithium might treat ALS. And we studied two groups of people. So the blue line, which you can just see there, is for people who weren't given lithium. They were given a drug that looks like lithium, but was actually nothing. It was placebo. And the red line is for people given lithium. And you can see it made no difference to survival. This is 3 different trials combined. There's no effect of survival from lithium. But if you break down the cohort by their genetic subtype, you can show that the people with this gene variation in UNC13A who were given lithium did much better than the people with the UNC13A gene who were just given a placebo. So that suggests that giving people with this genetic variation lithium might be a treatment. It won't cure them, but it might slow their disease down to make them survive more like a typical person with ALS, rather than the very aggressive disease they're more likely to have. And this is now the subject of a phase 3 trial about to start. The important thing is you could do this for any clinical trial, and I would suggest that all clinical trials from now on collect genetic data from our patients so that we can stratify them afterwards to see if there is a genetic subtype that might respond. The other question you can ask is whether molecular patterns map to the different clinical features of ALS. So neurologists can do this. So when I see patients, I might group them like this. This is my particular personal classification. And this is, again, a Kaplan-Meier curve. So the more to the right the line is, the better the survival. And you can see that people with the flail leg or flail arm type of ALS have a better survival than people, the black line, for example, with bulbar onset ALS. So my classification does predict survival to some extent. But you can give this same information to a computer and ask it, can you classify patients based on their clinical features to detect who will survive better or not? So we did that. This graph you can see on the right, the computer classified people without knowing their survival information. We withheld that from the computer. And you can see it made 5 groups, including this orange group where nobody died, and the red group, which has a very long survival with an average of about 180 months, so about 15 years. So that tells you that there is some underlying group or clustering of clinical features that does predict survival. It's just that neurologists and other humans aren't very good at detecting it, but it is there. We can also ask whether the survival effects I showed you, where some risk genes affect survival and others don't, might vary within a specific gene. And we've got enough variations in the SOD1 gene to answer exactly that question. So what you can see here is various SOD1 mutations that have been collected from the ALS online database, which you can access online. That's, you just type, it's just at alsod.ac.uk, alsod.ac.uk. If you go to that database, all this information is in there. And you can see survival on the y-axis. So, for example, on the left, we've got 2 very aggressive types of mutation of this gene, where people have a very short survival very consistently. Others where you consistently have a less aggressive survival, and some, like this big green block in the middle, where there doesn't seem to be any relationship between the gene mutation and how long you have ALS for. So even within the same gene, some variations don't connect with survival, they just increase your risk, while others increase your risk and affect your survival. So that suggests there might be different mechanisms even within the same gene. So it's a survival and risk connection or disconnection. Now, all of these things depend on knowing the mechanism, but actually we could do a treatment without knowing the mechanism if we can do a gene therapy. So here, we would simply target the actual mutation causing ALS. It would only be useful for the small group of people with a genetic form of ALS, unless we can find some way to make it more general. And we've got some initial studies where people have knocked down the SOD1 gene in people carrying an SOD1 mutation, and those initial results look promising. We're now in phase 3 studies. Now, I say they look promising. We've just had recently results from Huntington's disease, where a similar approach wasn't successful. But in another genetic disease, which is a childhood form of motor neuron disease called spinal muscular atrophy, these approaches work quite effectively. So we've got some hope that blocking the genes that are responsible for ALS in some people might be a good approach. There's also another study at the moment looking at knocking out C9orf72 as a gene when it's mutated. The question is whether we should also be looking at those common variations that only increase your risk a little bit. That might actually be important. For example, we know that in heart disease, even though cholesterol only gives you a small change in your risk of heart disease, giving a drug like a statin that lowers your cholesterol can have a big effect on the population. So it may be still worth treating even people with common genetic variations that only slightly increase your risk, rather than these genes that cause a family form of ALS. So just to conclude, we found many genetic causes of ALS. We're using those now to unravel the environmental risk factors, and we're just about beginning to find some consistent environmental risks. Understanding the causes of ALS is important for treatment if we know the loss of function pathways or the gain of function pathways. But actually, we don't necessarily need to know the causes if we have a genetic therapy for people with a genetic cause of their ALS. And the other thing we should always consider is this disconnection sometimes between risk and survival, because survival factors are also important since our patients come to us once they have the disease, and then prevention isn't such an important thing. So these are the sponsors of my research, but most importantly, this is my research team. These are the people who've done most of the work you've seen today. Thank you very much.
Thank you. Wonderful. I love it. Thank you so much. Can I ask you just one question?
Of course, please.
Okay. Yes, it's about thinking about people that are watching us. What message would you give them about the future, about the trials, okay, that we can expect it?
So I would say right now is the most exciting time in ALS research. I came into this field 27 years ago. At that time, we knew very little. We thought we knew a lot, but we actually knew very little. We didn't even have some of the scales we use today, like the ALS functional rating scale didn't exist. The ALS Scorial categories, which were a way of defining different groups of people with ALS, didn't exist. We knew nothing about changes of cognition or behavior in ALS. There was only one gene known to cause ALS, which was SOD1, and that had only just been discovered. There was only one treatment that had only just been discovered, which was riluzole. All of those things have massively changed. And also our knowledge of ALS is hugely increased. That's partly because we have very large international collaborations, which just didn't exist in those days. You know, now if I have an idea, I can have a call like this, or I can email my colleagues. In those days, my supervisor, Professor Nigel Lee, he used to send a fax and then wait for a fax to come back. That's how they communicated. You know, things are very, very much better now. And our knowledge of ALS means that we can do things much more. We can target which genes we think might, which treatments we think might be important for people. We can target them at those people. And we can also do those clinical trials better than ever before. So there are lots of new innovative ways of doing trials, but particularly exciting are platform trials, where you do lots of different studies at the same time. And there's at least 3 of those going on in the world at the moment. There's the Healy platform at Massachusetts General Hospital. There's the TRICALS initiative from Europe, we're part of that. And there's also the MND Smart platform in the UK. So all of those are trying to trial lots of different treatments at the same time. Because of course, if we only tested one drug a year, it's going to take us much longer to find a cure than if we test 10 or 20 drugs a year. So just having some system to do it very quickly is very important, but also understanding the causes and which drugs to use and which people to target them at is also very important. And I think all of those things are coming together now. So it really is the most exciting time in terms of ALS research. And I am hopeful that very soon we'll make a breakthrough, at least for some people with ALS.
Okay, great. Thank you. Thank you, Dr. Al-Chalabi. Thank you so much for being with us today. I hope to meet you next year. Who knows? Okay. Thank you so much. Bye-bye.
Thank you very much. And thank you very much for the invitation. It was very kind of you.
Ammar, thank you so much for giving us this fantastic lesson. It was an immense honor to have you here with us in Pro-Cura da Ela. I hope we can meet again in 2022. Thank you.
Thank you very much, both of you. I've really enjoyed giving the talk, and I hope to meet you again. I loved coming to Brazil last time I came.
Yeah.
Thank you.
Okay, thank you.
Bye.